Hey guys, I'm just going to go over the best answers on our review. Hopefully, if you've been watching everything and watching this, you feel really prepared going into the test. One, I fully admit, is kind of strangely worded, but I just wanted to make sure you had thought about this. So describe the difference between dominant and codominant in genetics. Okay, so if something is dominant, it will cover up another allele, okay? But if two things are codominant, then neither one covers up the other. If they're codominant, they each fully express their version of the trait, but in different areas. Okay, so if something's dominant over something else, the entire organism will show whatever the dominant allele tells it to do. Codominant, remember, they're going to kind of divide up and stake out their claim in different spots. So our examples we could talk about where it's like roan coloration, or in some flowers show codominance where like red plus white equals red and white, right? Red petals and white petals, or you might see red and white stripes or something like that. So that was a long answer to number one. Two, if one parent is heterozygous for blood type and is type A, the other parent is heterozygous for blood type and is type B, what's the probability their children will have type O blood? Alright, so the first parent is type A. So if you look back in the notes to see those alleles, you would see that the type A allele is like that, but it says they're heterozygous. So their other allele must be the recessive one that's covered up. Okay, second parent, same idea, except blood type B, but they're heterozygous as well. So we fill out our Punnett square, and I know the superscripts maybe weren't that easy to do, so don't worry if you didn't get that figured out. And it says, what's the probability their children will have type O blood? So remember, O is kind of for open. It means it doesn't have anything else. So that would just be this box right here. So hopefully you put yours in a complete sentence, but I'm trying to make time. 25% of those offspring would be blood type O, theoretically. Alright, why might two genes be linked? The only reason genes are linked is if they are on the same chromosome. Okay, anytime they're on the same chromosome, they're linked. They are likely to be passed together. But remember, they could still be separated during that crossing over process. So if this is one chromosome, and here's the homologous chromosome from the other parent, remember we have crossing over. So after crossing over, maybe they still got separated, right? On the right side of that chromosome, oh, those colors don't show up as nicely as I thought they would. That's a little better. So on this side of this one, it got a little bit of the green from that one. And this other chromosome got a little bit of the purple. So remember, for the linkage, they are more linked or more tightly linked if they are not only on the same chromosome, but close together. Okay? So what might cause two genes to be linked? They have to be on the same chromosome. But if you have, I'll try and draw a big one here. Okay. So maybe here's my chromosome. If I have a gene here and a gene here and a different gene way over here, okay, so crossing over can happen anywhere. Okay? A and B are really close together, so the likelihood that crossing over is going to happen right here is pretty small. Whereas the likelihood that crossing over is going to happen anywhere along this whole length between A and C is pretty good. So we would say A and B will be more closely linked, meaning they're less likely to get separated than A and C. A and C are still linked because they're on the same chromosome, but they're just less linked. They're more likely to get separated than A and B would be. Let me back up again. Alright. For homologous chromosomes, I just drew some right there. Okay, they're not necessarily identical, right? I get a little bit different instructions from mom than I got from dad. And if they had all the same alleles, that would make them identical, right? So they don't necessarily have the exact same alleles, but they should have the same genes in the same places. So A is your best answer for number 4. 5, phenotype is a combination of alleles that an organism has, whereas genotype is its appearance. Is that a true statement or a false statement? So remember, we say phenotype is for the physical appearance, and genotype is the genes, or what alleles they have. So this is exactly backwards, right? So phenotype is the appearance, genotype is the allele combination. So our best answer for that one would definitely be false. 6, a homozygous organism. And we're talking about homozygous. We're looking at things like this that have 2 identical alleles. Okay. So I'm looking back at my choices, and I actually kind of don't like this. So I probably will be a little bit leaning on this one. So this is really... Oh, I really messed up here. So this is a good answer. This would also be a good answer, which means that that's kind of messed up. I bet that I was going for D when I wrote this, but I'll accept B, C, or D. Let's talk about why A is not quite right. All right. So it has the same alleles for the same trait. All right. So if we think about one of our polygenic traits that we talked about, like skin color, right? So there's like 6 genes at least that help determine the skin color. In each case, the darker is more dominant. So for this first gene for skin color, I could be, well, I probably am lots of lowercase d's on this, but let's pretend this is me, Roxana Fees here. Then 1s and 2s just say which gene we're talking about. Okay, so these would be the 6 genes working together for skin color. That's one trait. Remember, I just made these up, but if this was true, I would say I'm homozygous for that one, homozygous for that one, but heterozygous for that one. So to say for a whole trait is sometimes not going to be quite correct. Okay, so I can clearly see that I kind of messed this up here. So I'll accept either B, C, or D on that one, but I kind of messed up. But they will produce gametes that carry all the same alleles. So if this is my genotype, I only have capital A's to give, right? So every single one of those eggs is going to have a capital A. Or if I was homozygous like this, every single one of them is going to have a lowercase a. So they're all going to have the same allele in every single gamete because that's the only allele that person had. Okay. All right, 7, segregation of alleles happens during meiosis. That is when we are creating our gametes. We're going to separate this A from that A, or that A from that A, whatever. All right. 8, the sex of a human is determined by the number of chromosomes they have. We all have 46. I shouldn't say, well, remember, there's a few chromosome disorders where you have slightly more or less, but generally we have 46. And so it's not the number of chromosomes. Remember, it's the type of sex chromosomes that you have. Right? If you have 2 X's, that's going to be female, and if you have XY, that's generally going to be male. All right, 9, genes that are found only on the X chromosome consistently show. Okay, so we just kind of talked about, remember, X's have to do with gender, so that's going to be our sex-linked inheritance. And remember, people associate the X with female, but X-linked traits, remember, are more likely to show up in males because even if, you know, this one has the instructions for some disorder, a female would have kind of a backup. Okay, so a female is going to have one good set of instructions. They're going to be able to be what's called a carrier, where they have one of those alleles for the disorder, but they don't show the disorder because they have this backup X. But a male with one kind of bad X, or an X with a allele for some disorder, is going to have it because he doesn't have a backup. So it is sex-linked inheritance. I just kind of don't want to talk about that as a quick refresh there. Dihybrid crosses. Okay, so in our notes we have, remember, dihybrid crosses are when instead of a simple Punnett square, we have to go with the big giant 4x4 here. And we might say we're looking at, what if we crossed parent that was this genotype with this genotype, right? That's kind of what we're looking at with the dihybrid crosses. So let's just see which of those descriptions matches what we're talking about here. Okay, so I have choice A, follow 2 alleles for 1 gene. So I have a gene that we use A's to represent, and a separate gene that we use B's to represent. So it can't be choice 1 or choice A because we have at least 2 genes. Follow the alleles for 2 genes. I've got the A gene and the B gene. A gene, B gene. That one looks pretty good so far. Looks at 4 genes. Nope, I just have the A gene and the B gene. That doesn't make sense. So our best choice has got to be B. Remember when you go to make figure out what the allele combinations would be in the gametes, that's when we do our first outside, inside, last we FOIL, and that tells us what the possible combinations in the gametes from that parent would be. All right, 11, Mendelian principles apply when genes are found close to each other on the same chromosome. Okay, so remember Mendel's laws of inheritance included the law of independent assortment. Which said genes do not affect each other. Okay, what do we just talk about when we're talking about genes found close to each other on the same chromosome? We were talking about gene linkage. Okay, so these are really kind of opposite. So Mendel said all genes assort independently. If they're on the same chromosome, they're linked. So that's going to be false. All right, dominant alleles mask what kind of alleles in heterozygous organisms? They are only able to mask those that are recessive. All right, the place where a certain trait's information is in the DNA. This is the specific version, and this is a whole chromosome, so it's got to be a gene. All right, 14. This describes a scenario where 2 different versions of a trait both show up in different places. So again, this is like red flower plus white flower equals red and white flower. Maybe it has red and white petals, or something like that. Or, you know, an allele for type A blood and an allele for type B blood makes AB blood. Right, so this was the co-dominant. And this one is the one where instead of having divide and conquer, it's a blend. We have an in-between form. Okay, so red and white would make what's in between red and white pink, right? Or, trying to think of another really good example. That's kind of the classic one. But when we have an in-between form, or there's another one, black and white, if we have an allele for black and an allele for white, in between would be gray, right? So that is incomplete dominance, where you have the heterozygote is an in-between the 2 extremes. All right, and last question. Things like skin color display a wide range of possible phenotypes because their instructions are spread across multiple genes, each with different possible allele combinations. I just talked about skin color. Okay, so we've got these different genes that each have instructions that contribute to one trait. Okay, and this word literally means many genes, right? Poly for many, genic for genes. So if you have anything that's described as having a wide range of possible phenotypes, it can have that because we have lots of sets of instructions all kind of contributing a little bit to the final end result. All right, test should open up at noon on Thursday the 16th. Let me know if you have any questions before that. Good luck. Thank you.